This is module A8, objective 2, and in this objective we're going to look at the special sense of gustation or taste. Gustation allows us to perceive information about the foods that we eat and the liquids that we drink. The particular receptor type are called taste receptors or gustatory receptors. And we find that gustatory receptors are localized on the tongue. They're found in the pharynx, and the pharynx ranges from this region here. And they're also found down in the larynx, right here. When we look at these receptor cells, we find that they are found in taste buds. And a taste bud would be receptor cells with associated epithelial cells, sometimes epithelial projections, that are called lingual papillae. And taste buds are found predominantly on the superior surface of the tongue. Of the lingual papillae, there are three types. The first is the filiform papillae. Filiform papillae do not contain taste buds. They do nothing for taste. Instead, they provide surface area for friction. We've all felt a cat's tongue, how rough a cat's tongue is. And again, it's the friction that is being caused by the projections on the cat's tongue. And we also have similar projections. These are the filiform papillae. And the role or the function of the filiform papillae are to help grasp the food and move it towards the back of our mouths. Fungiform papillae, fungiform because it looks like a small mushroom cap, funga for fungi. Fungiform papillae contain a small number of taste buds, upwards of maybe 5. And again, they're distributed in various regions of the tongue. The last type of papillae are the circumvallate papillae. And the circumvallate papillae contain numerous taste buds, upwards of 100 taste buds. And we find that the circumvallate papillae are somewhat localized on the superior surface at the rear of the tongue, or the inferior portion of the tongue, posterior portion rather, in a V-like shape. When we look at a taste bud, we find several cell types. We have a basal or stem cell. Again, basal or stem cells are there to replace receptor cells that are past their due date, so to speak. We have the receptor cells themselves, the gustatory cells. These gustatory cells last about 10 days, so the basal cells are very active in replacing gustatory cells. When we look at the gustatory cell, we see that little microvilli extend off the distal portion of the cell, and these are referred to as taste hairs. And taste hairs are going to be open to the environment through a structure known as the taste pore. And we see here's the boundaries of the taste pore. So this would be the surface of the tongue, for instance. We know that gustatory cells are monitored by a number of cranial nerves. If you think back to a week or so ago, you should be able to identify the facial, the glossopharyngeal, and the vagus nerve as all being sensory nerves carrying fibers from the tongue. And those cranial nerves are going to synapse within the medulla oblongata, and from there they'll head to the thalamus, and then lastly to the primary sensory cortex, where we actually perceive the taste. So again, when we look at gustatory discrimination, there are a number of primary taste sensations: sweet, sour, salty, and bitter. In addition to these, your textbook will mention two others. Umami is a taste that we associate with meat, for instance. Beef, chicken broths, Parmesan cheese. These particular receptors are sensitive to small proteins, small peptides, individual amino acids. We also have water receptors. And I know people assume that water has no taste, but we actually do have receptors that are specific for water. And these receptors tend to be localized in the pharynx. If you think the back of your mouth as the region of the pharynx, this is where we're going to find many of the water receptors. In terms of discriminating tastes, the process involves, again, dissolving chemicals in some type of liquid medium. As we take food or some type of liquid into our mouths, those chemicals within the food product or the liquid will dissolve within our saliva, and those molecules will make contact with the taste hairs, which are full of receptors. They'll bind to the receptor proteins, and of course, if that binding is strong enough, it initiates an action potential that is, again, sent to the brain for processing. How, with only a handful of different taste sensations, can we discriminate between the thousands of tastes that we've experienced in our lives? Again, it's the pattern and frequency of receptor activation. It's the pattern and the frequency of those action potentials as they're being shunted back to the cortex that ultimately translates into a particular taste. End results. With the binding of the receptor, there's a release of the neurotransmitter, and then you get the generation of an action potential in an afferent fiber heading back to the central nervous system. In terms of sensitivities, we find that people are different. Some people taste things differently than others. Some of these differences are inherited. For instance, there's a well-known case involving phenylthiocarbamide. I'm sure many of you incorporate that into your evening meals on a regular basis. It's found that 70% of Caucasians can taste it, while about 30% cannot. So there are some differences, and they can be inherited. The number of taste buds that we have starts to decline, and they decline rapidly by around the age of 50. Again, hearing your grandparents complain about how things don't taste the way they used to. Well, of course not. You're losing your taste buds.